If you're interested in microbiome, you're probably aware of how rapidly the field of microbiome research is evolving these days. What you may not have heard of is that microbiota testing can now be performed in a fully standardized way in any diagnostic laboratory. My name is Zuzanna. I am a biotechnologist specializing in molecular microbiology, and for the last 4 years I have worked with assay development at genetic analysis in Oslo. And at Genetic Analysis, we are a research-driven diagnostic company at the very forefront of microbiota diagnostics. In this presentation, I want to talk to you about how we at Genetic Analysis standardized high multiplex microbiota testing with help of Luminex's xMAP technology. And although it is a complex subject, I will break it down to you to my best ability. So let's start by introducing you to our field of expertise. What is microbiota? Microbiota is all of the microorganisms like bacteria, viruses, and fungi that inhabit different sites on the human body. Bacteria are the most widely studied group, and they also make up the biggest mass of the total microbiota. So just to clarify going forward, when I refer to microbiota, I refer to the bacteria. And the microbiota is something we all share, and to a large degree, we are all dependent on its existence. So you may have heard terms like gut microbiota or oral or skin microbiota, which are used to collectively describe distinct composition of microbes on different body sites. Here I want to mostly focus on the gut, which is home to the body's largest and arguably most important collection of microbes. Gut with its inhabitants is a very interesting place. The gut is many meters long, and because of the huge total surface, it's home to millions of bacteria. But even though the gut is placed deep inside the body, it actually comes in contact with the outside world all the time. If you think about it, when you eat food, you introduce the outside world to the inside of your body with all benefits, but also all potential threats that go with it. So, in a sense, gut and gut microbiota are sort of a frontier between the outside and the inside. Because not only is the gut connected to the outside, it is also connected to the rest of the body to be able to feed nutrients extracted from the food and the bacteria-produced metabolites to the body. One of the most important internal connections is what we call the gut-brain axis, or sometimes even microbiota gut-brain axis, which form physical and physiological channels of communication between the gut and the brain. Now, keeping all that in mind, it is much easier to understand the role and the importance of the gut microbiota. Among the bacteria's many functions, the most crucial ones include obviously help with digestion by breaking down complex carbohydrates, and in breaking down the food, not only do the bacteria provide nutrients, but also produce different compounds like metabolites which would otherwise be unreachable for us. Being on the frontier of outside and inside world, the bacteria also provide sort of a barrier protecting us from any pathogens and toxins that may come with the ingested food. And last but not least, many bacteria hold an anti-inflammatory function crucial to the body's defenses. And only these few mentioned functions make up quite an impressive resume for something seemingly invisible. Now, remember that gut bacteria form a complex network of many different species that exist in a specific balance. And you can think of it as an ecosystem. And as it is with ecosystems, they vary in terms of types and variety of species present. And such is also the case of gut microbiota as an ecosystem, which varies from individual to individual. But as long as the species composition is in balance, so the crucial actors are present and the diversity is retained, the ecosystem thrives and it can perform all of its vital functions. However, if this balance is lost, the functions will be impaired. And for gut microbiota, we use the term dysbiosis, which is a state of imbalance characterized by species depletion, loss of diversity, and overgrowth of certain species. Dysbiosis is a term you want to keep in mind. So intuitively, you would connect dysbiosis to gastrointestinal disorders like irritable bowel syndrome or inflammatory bowel disease, and you would be right. But yet, more and more studies confirm that dysbiosis is associated with a whole range of diseases and disorders affecting the whole body, ranging from diabetes to cancer to cognitive disorders and mood disorders. And considering the gut microbiota's connection to the rest of the body, it really is no wonder that its association with all of these disorders is rapidly gaining interest. And now gut microbiota stands in the center of possibilities for both diagnostic values and targeted treatment options. So, what is stopping us from integrating microbiota testing into diagnostic procedures and treatment development procedures? Well, traditionally, standard microbiota testing allows for looking at one species at a time, often with pathogens in focus. But testing for multiple single species or strains separately does not give an idea of the overall composition and balance of the gut microbiota. And this makes multiplexity the first hurdle. Another challenge is standardization. Considering the differences in microbiota composition between individuals and between communities around the globe, there is no such thing as one perfect microbiota. So there is really no one-size-fits-all solution. And here, allow me to tell you a story of how we at Genetic Analysis managed to overcome both of these obstacles. And our story starts with the DNA. So when genetic analysis started off years ago, we wanted not only to look at multiple bacteria simultaneously, but also to be able to tell if the microbiota composition is in balance or not. And at this point, we were already able to identify different bacteria groups with help of this gene, the 16S rRNA gene. 16S is a housekeeping gene, meaning that all bacteria have it, and parts of the gene called the conserved regions are largely the same for all bacteria. And this means that by targeting this gene in PCR amplification, we target all the bacteria present in the sample. What is also very useful about the 16S gene is that it also has numerous variable regions which are different for different bacteria groups, and they allow us to identify these groups based on a specific sequence within a variable region. So using this knowledge, we designed a set of DNA probes, which are short oligos complementary to the target bacterial DNA sequence. The probes targeted different bacteria, from single bacteria species to larger groups of bacteria, but what they all had in common is all these bacteria were known from peer-reviewed studies to play important roles in the gut environment. The 48 probes selected for our panel were chosen based on their discriminatory power to distinguish healthy from dysbiotic microbiota. But even though it's just 48 probes, together they target over 300 different bacteria species. So how it works in a reaction, when adding our probe set to the amplified gDNA from a fecal sample, each probe will find its target if it's present in the sample and anneal to it. We also add DDCTP biotin complex to the reaction, and the probes whose target sequence is present will be extended with a DDCTP and thereby labeled with biotin. And this will only be the probes whose intended target sequence is present because the target sequence will have a G complementary to the DDCTP. And this little tweak makes the probes highly specific, and at the same time, it is a very simple way to be able to tell with high certainty what is actually there in a sample. But this is only a partial success. To be able to convert the reaction into readable results, we utilized Luminex's xMAP technology. Another set of short DNA probes, and this time amino-modified probes, complementary to our labeling probes, was coupled to multiplex microspheres, one probe type per region. In a hybridization step, all complementary probes will bind to their corresponding probe coupled to the microsphere, and then when we add a fluorophore to the reaction, only the biotin labeled probes will have a binding seat for the fluorophore. With the use of Luminex 200 instrument or MagPix instrument, this fluorescence is converted into signal values. And as you know, the instrument identifies the microsphere or the region, and then also the fluorescence signal of the probes attached to it. Signal strength corresponds to the abundance of the target in the sample, and that makes it possible to not only detect the presence of the target, but also to define how abundant it is. And in short, this is how the xMAP platform works. But of course, we wanted not only to detect bacteria, but also to differentiate a healthy microbiota from a state of dysbiosis. And to achieve that, we approach the problem as you would do in any diagnostic test development. We collected data from healthy individuals across Scandinavia to create a healthy microbiota model, while cohorts of IBS and IBD patients were recruited to find the cutoff thresholds. Based on the microbiota patterns detected with our probe panel, we were able to create a healthy reference model and develop an algorithm. How the algorithm works is if a sample falls within the established limits for the healthy population, it is considered healthy. However, if a sample differs from our reference group, it will be considered dysbiotic. And we did not stop at Scandinavia. Our mission was always to bring standardized microbiota testing to the world. So in the following years, we introduced cohorts from different sites in Europe and North America, and we validated the test for use across Europe and United States and Canada. And this clinical validation work is constantly ongoing. It's this open project, and as we speak, we work with our partners in different countries like China and Thailand, to name some examples, to further validate the test. If you are not convinced that our model should work globally, here is something to support our work. A few years back, in an industrial PhD project, which was a collaboration between the Norwegian University of Life Science and Genetic Analysis, we created this gut bacteria genome database called HAMGUT. And HAMGUT is based on shotgun metagenomics data, so a completely different method to ours. And it collects around 30,000 bacterial genomes from different sites around the world. Now, when you look at these plots generated from HAMGUT, you will find that there is this almost perfect overlap of species that you can expect to find in a healthy gut, whether you're in Denmark or Spain or China or USA, and you would actually have to travel to geographically isolated locations and maybe more indigenous communities to find microbiota profiles that significantly differ from our reference. So here we are. The xMAP platform and the xMAP dysbiosis test really opened the possibility for standardized multiplex microbiota testing. Today, xMAP dysbiosis test is a CE-IVD labeled test used in laboratories globally, and it is available on the market as a reagent kit. By feeding the Luminex signal data to our own software, which contains the algorithm, what you end up with is a report pinpointing the sample's dysbiosis status, as well as the status of some crucial functional bacteria groups, and information about the abundance of each marker compared to the healthy reference group. The whole process takes under 2 working days, and thanks to the integrated software, it requires no data processing steps. Clinical validation work that we do continuously illustrates one aspect of standardization. In spite of its complexity, treating the microbiota like any other parameter and using clinical data to develop models and thresholds allows us to compare cohorts regardless of location. And on the other hand, our approach of pre-selected marker panel is actually crucial to the success of this method because it narrows the field of focus to what we already know about the microbiota rather than exploring single species that differ between communities or locations. But there is a duality to standardization, and the second aspect is our use of the xMAP technology in the multiplex assay development. Anchoring the assay on an established hybridization and detection technology is also what contributed to a standardized workflow of our platform. And this strategy led to many benefits, from streamlining production to increasing throughput to achieving excellent robustness and, most of all, expanding accessibility of the method, because every Luminex user would be able to take our method into use. And having created this platform, we were never going to just stop there. The world of microbiota, as I mentioned before, still holds a great potential, whether you're looking for biomarkers, disease mechanisms, or treatment targets. With our newest developments, we wanted to offer a robust and standardized tool for microbiota research that would be suitable for both academia and pharma, while still retaining the simplicity of our method and drawing benefits from the high multiplexity potential offered by Luminex's xMAP technology. And we called our newest research tool the xMAP Discovery. So this time, taking into use all of the information that is available about bacteria that are associated with gastrointestinal and metabolic health and disease, we went through numerous peer-reviewed publications and exploratory studies, and we put together a panel of over 170 DNA probes, targeting all these different bacteria groups, but this time coming from both oral and gut microbiota. We documented the specificity of each marker through both in silico and in vitro testing on bacterial gDNA. We also tested precision, so both repeatability and reproducibility of the markers, and we were happy to see that the assay performs just as well as expected and just as well as our previous panel, showing little variation between replicates within run and between runs. And putting together this wide range of markers, we treat it as an open invitation to explore, because we believe that this assay can have so many applications in academia and industry research. So whether it's comparative studies or different disease cohorts, treatment response studies, microbiota diversity studies, it is a huge advantage to use a standardized and documented tool that is not dependent on external databases for bacteria identification. But of course, don't take my word for it. xMAP Discovery's primary purpose is to be able to compare and discover differences in different patient groups or sample types based on their microbiota composition. So the first and obvious test was putting together markers from both oral and gut microbiota. We wanted to see if the assay will be able to differentiate samples coming from these different sites. And as you can see with this PCA plot of fecal and oral samples, they separate perfectly based on their signal patterns, as they should. But as the last point, I want to share with you our own example of how we implement use of xMAP discovery in our own assay development pipeline. 2 years ago, we started a new project to develop a companion diagnostic test for disease course prediction in IBD patients. The project received funding from the Research Council of Norway, and the goal is to identify bacteria signatures that associate with mild or moderate or severe disease course at a diagnosis point or at the point of treatment escalation. And in this context, we use the xMAP discovery panel to look for these bacteria signatures. While we're looking to start the clinical studies shortly, in the meantime, we tested the panel using anonymized patient samples from our biobank at Genetic Analysis, and the cohorts included IBD, IBS, and healthy patients. And as you can see, using the panel, we managed to achieve separation between these three groups, while we also managed to extract specific biomarkers that played a major role in differentiating between these groups. Here I'm showing just a few from the list, while we managed to actually find around 50 for these cohorts. So if I dare to look a little bit into the future, as microbiota is gaining interest as both treatment target and potential biomarker pool, a need for standardized microbiota testing, microbiota-based screening tools, and companion diagnostic assays is growing. In this setting, our continuous work to standardize microbiota testing while we utilize the potential and availability of xMAP technology paves way for such assays and shortens time from biomarker discovery or microbiota pattern discovery to implementing such assays into standard procedures. And with that, also with big smile and greetings from our team at Genetic Analysis, thank you for your attention.